Mitosis is the process of cell division after DNA synthesis meant for creating two identical cells. Mitosis operates in process of growth and repair in organisms.
Interphase is the longest part of a cell's life and is not even considered part of the reproductive phases, such as mitosis or meiosis. Interphase is just the state the cell is in when not reproducing. Two centrosomes are present here in the color yellow. The nucleolus is in brown, the chromatin is blue, and the nuclear envelope is in purple.
During prophase, the first phase of mitosis, the genetic material, or chromatin, begins to condense in the nucleus. The nucleolus begins to disappear, and the nuclear envelope begins to dissolve.
Prometaphase is the small mitotic phase in between the two larger pro and metaphases. During this phase, the chromatin begin to form chromosomes in sister pairs, called chromatids. Two chromatids make up a chromosome. The nuclear envelope disappears and mitotic spindles begin to form.
The formation of spindles is complete, and microtubules, meant to aid in movement, are attached to the chromosomes. The microtubules attach to the kinetochore of each chromosome. The kinetochore is plated out proteins meant as an attachment site for the microtubule.
In anaphase, the identical chromatids in each chromosome separate, as each half is meant for one cell. The microtubules attached to each chromatid begin to shorten, causing a separation in genetic material directed towards each pole where the centrosomes are located.
In telophase, the cell begins to divide, forming two daughter cells. Simultaneously, a process called cytokinesis takes place, where a division in the cell happens along the cleavage furrow. At the end of mitosis, the genetic material is left in the form of 2n, where the number of chromosomes is equal to the number that we began with, making two diploid cells.
Meiosis is a process of two cell divisions after DNA synthesis that creates four similar but slightly different genetic cells. Meiosis operates in the process of sexual reproduction.
In prophase 1 of meiosis, chromatin condenses into chromosomes and they pair themselves with another chromosome into a state called synapsis. Here a phenomenon called crossing over takes place, where the chromosomes switch partial genetic material. Crossing over is one of the factors that adds to genetic variability.
In metaphase 1, homologous chromosomes line up at the metaphase plate of the cell. Microtubules attach to the kinetochore of each chromosome. In anaphase 1, microtubules pull towards the poles and one chromosome of each homologous pair moves towards the cell's pole.
In telophase 1, chromosomes are at either pole, and the cleavage furrow forms, separating the two cells.
Spindles form in prophase 2 and chromosomes move towards the metaphase plate.
In metaphase 2, the chromosomes are on the metaphase plate, but the sister chromatids are slightly different due to crossing over and independent assortment in prophase 1.
In anaphase 2, chromatids move towards the opposite poles after the chromosomes are pulled apart.
In telophase 2, the cells divide. 4 cells are left over with different genetic material, different from one another and different from the parent cell. Each cell is left with half the genetic material of the parent cell, and they are termed haploid. The differences between mitosis and meiosis are: in mitosis there is one division, while in meiosis there are two divisions. In mitosis the cells are exactly the same, and in meiosis there is genetic variability. Mitosis is used for the functions of growth and repair, while meiosis is used for sexual reproduction. The end result cells of mitosis are diploid, while the end cells of meiosis are haploid.